I warmly welcome Doris Bachtrog for our first talk. So I don't think I need to introduce her. She's a professor at UC Berkeley, so one of the most influential scientists working on the evolution of sex chromosomes and their evolution, how they are formed, how they degenerate. And she will talk today about the toxic Y chromosome and their degeneration. Thank you very much.
Thank you very much. Okay, first of all, it's a pleasure to be here at this meeting, and thank you very much for organizing it. So today I'll be talking about toxic Y chromosomes.
Does it work?
I think so, yeah.
Okay,
yeah. And
sorry.
the point there, yeah. Okay, so in many species where we have males and females, sex is determined by a pair of heteromorphic sex chromosomes, which in addition to their sex-determining role have evolved independently a series of unique features, as everybody knows here. So X chromosomes typically are gene-rich, or they resemble gene-rich autosomes. They're full of genes, they have few repeats. Y chromosomes, on the other hand, typically only have, or old Y chromosomes have very few genes left, and they often have acquired a large number of repeats. Interestingly yet, sex chromosomes have evolved independently many times from autosomes, which means that initially they had the same set of genes, so X and Y chromosomes both were gene-rich autosomes. And sex chromosomes have evolved not only once, but as everybody knows here, they have evolved dozens of times in plants and hundreds of times in animals. We know a lot about how sex chromosomes have evolved, so we know that sex chromosome differentiation is mainly driven by a progressive loss of genes on the Y chromosome, and simultaneously Y chromosomes accumulate repetitive DNA. And we also know that one of the main reasons why sex chromosomes differentiate is because there's a lack of recombination between the X and the Y chromosome, which allows them to evolve independently. And so my lab has been using for decades now neo-sex chromosomes of Drosophila, of the genus Drosophila, to try to infer the evolutionary history of how an autosome transitions into a sex chromosome. And so those neo-sex chromosomes are formed by just ordinary chromosomal fusions between ancestral sex chromosomes and autosomes, and over time, those fused autosomes that don't recombine with each other, they start differentiating into heteromorphic neo-X and neo-Y chromosomes. And so in the past, like 5 or 6 years, we have spent a lot of effort trying to develop sequencing approaches to actually be able to fully sequence those diverging and heteromorphic sex chromosomes. And so in the past couple of years, we have actually acquired whole genome sequences of Y chromosomes from about a dozen different Drosophila species that have neo-Y chromosomes that were formed at different time periods, so some very young ones, some very much older ones. And so that sort of general finding or the general pattern that we find is that at least in Drosophila, a young sex chromosome is anything that's less than 100,000 years old. And in those cases, those young sex chromosomes, they basically resemble an autosome. So you have a lot of active genes that are transcribed. There's just a handful of pseudogenes accumulating. On the other hand, once you pass 10 million years or so, the Y chromosome or the neo-Y chromosome basically resembles an old Y chromosome. So we have a lot of transposable elements, a lot of satellite DNA, a lot of repetitive junk DNA, very few genes. Those old neo-Y chromosomes are typically entirely heterochromatic, so that whatever junk there is being epigenetically silenced. And then we have these intermediately aged sex chromosomes or neo-sex chromosomes, which is about 1 to 10 million years, where we have this assemblage of having still a lot of active genes on Y chromosomes, but they start degenerating, yet we also see a dramatic accumulation of repetitive junk DNA. And so one of my favorite species over the last few decades has been Drosophila miranda, where a neo-sex chromosome formed about 1 million years ago, or 1.5 million years. And in this species, this just shows you a snapshot of the neo-X and the neo-Y chromosomes. So genes are color-coded. So we still have almost all of the genes of the ancestral genes remaining on the neo-Y chromosome. But those genes on the neo-Y, they're actually being embedded by large chunks of repetitive DNA. So in this particular case, the neo-Y chromosome is actually 4 times as large as the neo-X chromosome. It has a similar number of genes. It actually has slightly more, but it has huge levels of junk DNA. And so in today's talk, I will be talking about the idea that repetitive Y chromosomes can actually be toxic. And second part of my talk, I will be talking about this idea that, especially in this intermediate sex chromosomes where you have actively transcribed genes interspersed with repetitive junk DNA, you could actually have an epigenetic conflict between the need to repress junk, yet also needing to transcribe active housekeeping genes. And so you could end up in some sort of epigenetic conflict here in toxic Y chromosomes. And in the first part, I'm telling you that even if you have a very old Y chromosome where all you have left is repetitive junk DNA, so no transcribed genes, you still can have a problem in males, and you can have an epigenetic conflict between males and females. Since males, if you have a large heterochromatic Y chromosome, you will have much more repetitive DNA, and so this could actually have an influence on the dynamics of heterochromatin between the sexes. And all the work I'm talking about has been done by 3 very talented grad students, Lauren, Allison, and Emily, and the postdoc, Kevin. So repetitive junk DNA, as I mentioned, is typically silenced by heterochromatin, but there's 2 stages in the life of a fly when heterochromatin formation is somewhat compromised. The first one is during very early development when heterochromatin is first being laid down on repetitive regions to silence them. And the second one is late in life. So it has been shown that late in life in many organisms, heterochromatin starts to deteriorate, and this has been shown, across animals. Like heterochromatin loss has been found in humans, it has been found in yeast, it has been found in Drosophila, and concordant, as I mentioned, like heterochromatin is used to epigenetically silence repetitive DNA. As heterochromatin is becoming lost, as organisms age, transposable elements are becoming reactivated, and this has led to this model of heterochromatin associated. A model of senescence. So early on in development, you have heterochromatin formed and it's found at the periphery of the nucleus and it silences repeats. But as organisms age, heterochromatin becomes disraveled, it deteriorates, transposable element can become active, and this can lead to cellular senescence. So as I mentioned, the amount of heterochromatin can differ between males and females. So males, at least in mammals and Drosophila, males have more heterochromatin. Heterochromatin is getting lost during development or during aging. Interestingly, in many species, there's also a difference in longevity between males and females. So what I'm showing you here, and I'll show you many of those plots, are survivorship plots where on the y-axis is the fraction of the population surviving, and on the x-axis is age. And this shows data for humans at 2 different historic time points. And for each historic time points, females tend to live longer than males. And what's true for humans is true for the elephant, and is true for a large number of Drosophila species. So in general, males seem to be living longer than females, but that's not a general pattern. It's not always males outliving females. So there's some cases where males, in this case, the open circles, are actually living longer. But in this study, all the longer-lived males are birds. And everybody in this audience here knows that in birds, it's actually the females that has a Y chromosome. So this data already suggests that it might not be being a male that makes you live shorter, but the presence of heteromorphic sex chromosomes. And indeed, there were a couple of recent studies showing on a more global scale that the heterogametic sex in vertebrates or across animals tends to live shorter than the homogametic sex. So as I mentioned, heterochromatin amount differs between males and females, or between the heterogametic and the homogametic sex. The heterogametic sex tends to live shorter, and heterochromatin is being lost during aging. So what we were interested in is showing whether there's indeed some evidence that the sex-specific epigenome differs between males and females during aging, and whether a loss of heterochromatin might contribute to faster aging in males. And so in particular, we tried to answer 3 questions. First one is, does heterochromatin loss, does it occur faster in males than females? Do repeats become more de-repressed in old males compared to old females? And does a Y chromosome contribute to a reduction in lifespan? And we used Drosophila for answering those questions. So in Drosophila melanogaster, the males have this very large heterochromatic Y chromosome, and so overall about 40% of the male genome is heterochromatic, but only about 30% of the female genome, so there's this like overall difference. And indeed, in Drosophila, this is a survivorship curve, males live shorter than females. And so what we did is we sampled young and old males and females, and we collected both chromatin data, so using CHIP to measure heterochromatin across the genome, and also using RNA-seq to look at transposable element activity across aging in males and flies. And what I'm showing you here is the carrier type of Drosophila, so in gray is heterochromatic regions, and I'm just laying out the chromosomes, so around the pericentromeres you have heterochromatin, the Y chromosome is entirely heterochromatic. And so what I'm showing you here is heterochromatin enrichment for, in red it's females, and in blue it's males, and this is for young flies, so as expected we see a lot of heterochromatin around the pericentromere, which is highly heterochromatic. We see a lot of heterochromatin at the Y chromosome. Just to mention, the Y chromosome in melanogaster is fully assembled, so there's much more that we can't map because it hasn't been assembled. And what's important here is that we see heterochromatin at the pericentromeres, and it's very similar of how much for the young flies, and it's very similar between males and females. However, as flies age, we see that in females we actually sort of maintain the same amount of heterochromatin, so this is for 68 days, but in males we actually see that there's a dramatic loss in heterochromatin. And that's true for the pericentromeres, and it's true for the Y chromosome, and this is just using the same data but subtracting the signal from the old flies minus the young flies. So we see that there's actually a little bit of loss of heterochromatin in females in red, but there's a dramatic loss of heterochromatin in males. So loss of repressive marks in pericentromeric regions occur in males during aging, and it's more pronounced in males compared to females. So the next question we were interested in is whether this loss of heterochromatin also results in a de-repression of transposable elements. And so what I'm showing you here is expression data. So this is a heat map. Each row here shows you expression for a specific transposable element family. On the left is males, on the right is females, and this is for young flies. So as expected, most and the more red, the more high expressed. So as expected, transposable elements are efficiently suppressed in young flies. And as flies age, you can see that transposable elements are still efficiently suppressed in females, so this shows you the heat map. This is the same data just as a scatter plot, like expression in young versus old females, young versus old males. And as you can see, like females keep suppressing their transposable elements as they age. If anything, it becomes more efficient suppression. But in males, we see that many families of transposable elements start to become deregulated and start to become expressed more highly as the flies age. And if we look at transposable elements that are specifically located on the Y chromosome, we see that this increased expression of TEs is actually much more pronounced. So there's a dramatic increase of expression of transposable elements found on the Y chromosome. So this loss of repressive marks in males indeed seems to be resulting in increased repeat expression. And so the final question is whether we try to answer is whether the presence of a Y chromosome directly contributes to shorter longevity. And here we can take advantage of classical Drosophila genetics. So we can use this, they're called attached X and attached Y stalks, where the X and Y are actually fused, or two X chromosomes are fused. And we can just use like standard crossing to generate flies that either, so we can generate females, for example, that have an X chromosome or two X chromosomes, but also a Y chromosome. We can generate males that don't have a Y chromosome. So in Drosophila, sex is purely determined by the number of X chromosomes. The Y chromosome doesn't have a sex-determining role. So X-normed males, so to say, are males. And we can also make males that have two Y chromosomes. So here we can vary the number of Y chromosomes independently of sex. And we can put a Y chromosome in a female, and we can have 0, 1, 2 Y chromosomes in males. And then we just measure longevity of those different flies. So we take a large population of wild-type females, wild-type males, females that have a Y chromosome, males that don't have a Y chromosomes, and males that have two Y chromosomes. And we just take large populations and put them to new vials every couple of days and record the number of dead flies. So that's how you make those survivorship curves. And so this is the data for Canton S, which is a wild-type fly strain in Drosophila. And we used two different attached X and attached Y lines to make those crosses, to make those XXY and X Nord flies. And so I'm just showing you the 3 different replicas for the 3 different strains. And what I'm plotting below is just the 50% survival, so the number of days where 50% of the population has died. And as mentioned earlier, and as shown in these plots, males die significantly earlier than females in wild-type flies. If you look at the 3 different replicates for our attached X, attached Y crosses, in each case, if you have a female that has a Y chromosome, that female lives significantly shorter than a wild-type female. On the other hand, if you have males that don't have a Y chromosome, so the X Nord flies, and every time their survivorship, the longevity is significantly increased, it's much higher than wild-type males, it's actually higher than wild-type females in most cases. And finally, if you make males that have 2 Y chromosomes, those guys die very early on, so they have dramatically decreased survivorship. So this suggests that indeed it is the presence of a Y chromosome that directly contributes to a reduction in lifespan. So what I showed you so far is that as flies age, heterochromatin that's normally found at pericentromeres and the Y chromosome is becoming deteriorated, and it's being deteriorated more strongly on the Y chromosome, resulting in repeat activation in old males and contributing to shorter survivorship. The other time period that I told you where heterochromatin is compromised is in early development. So in fruit flies, the first couple of hours of development, so you start with an egg, and this is early embryogenesis. So most of the first 2 hours or so of development are fully driven by maternal transcript, so the embryo doesn't have any zygotic transcription yet. But about an hour into development, the zygote starts taking control of its own transcription, so you have global genome activation. And around the same time point when the genome is becoming activated, epigenetic marks are being laid down across the cell, including heterochromatin, to make sure that you suppress repetitive DNA. And so we decided to look at heterochromatin formation in early development, contrasting 2 species that have very different Y chromosomes. So the first one is Drosophila miranda, the other one is a close relative, Drosophila pseudo-obscura. And we chose those species for multiple reasons. So on one hand, they're closely related, so they have the same repeat families. We can compare the same transposable element families. And we also produced very well-assembled Y chromosomes for both of those species. On the other hand, they have a very different type of Y chromosome. So Drosophila miranda, as I mentioned, has this young neo-Y chromosome where we have thousands of genes interspersed with repetitive DNA, versus Drosophila pseudo-obscura has a more traditional old Y chromosome where there's almost no genes remaining, just a handful of genes, and most of the Y chromosome is repetitive chunk DNA. So the miranda is a youngish Y versus pseudo-obscura is this traditional old Y chromosome. And so we looked at gene expression during this very early development in individual female and male embryos. So this is just averages of female and male embryos. And what I'm showing you here again is a heat map for expression of individual transposable element families in females versus male across development. And this arrow here indicates when the zygotic genome is becoming activated. So the first 2 stages here, we only see maternal transcripts. And then once the arrow starts, we start having the zygotic genome activated. And this is transposable element transcription for miranda, and this is for pseudo-obscura. And as you can see, once the genome becomes activated, we start seeing that there's a differentiated transcription between males and females, and more transposable element transcription in males than there is in females for both species. And this is just replotting the same data, just adding up just all the number of transposable element transcripts across families. In blue is males and red is females. So as the zygotic genome becomes activated, there's a burst of transposable element transcription, especially in males, and this burst continues across development in miranda, and there's a short burst in Drosophila pseudo-obscura where there's substantially more transposable element transcripts in males compared to females, but then it levels out later in development. And we can divide transposable element into those that are found on the autosomes or the X chromosome versus those that are Y-linked. And I'm showing you here across development female expression, male expression. And so the main point of this slide is showing that there's a little bit of a difference for expression between males and females for autosomal and X-linked transposable elements, but it's not very dramatic. In contrast, if you look at Y-linked TEs, those are the ones that are highly upregulated in males. So there's like this dramatic burst of expression of Y-linked TEs during early development. So there's a burst of TE expression. The next question we were interested in is, does this burst in TE expression during early development also result in more TE insertions, so in more somatic mutations as males develop? And to do that, we use a sequencing approach. So we sequence early embryos, and we can, just like individual embryos, and we can identify novel insertions by mapping paired end reads to the genome and to a TE library. And if we have shared insertions, then we will find a particular insertion found in all individuals, and if there's de novo insertions, then it's an insertion that's only found in a particular embryo. And these here are data, like each dot here is a specific embryo. In blue, it's males, and red, orange is females. What I'm showing you here is the number of unique insertions inferred for each of those embryos, and it's plotted against sequencing depth, so the more we sequence, the more insertions we can identify. And here I'm just showing you the residuals from this regression line. And as you can see, there's significantly more insertions that we infer from male embryos compared to female embryos. And that finding is significant for Drosophila miranda, but not for Drosophila pseudoobscura. So in Miranda, where we have this widespread burst of TE expression, we also see more TE insertions in males compared to females. And as expected, if we look at which families of transposable elements show more insertions, it's the ones that are higher expressed in early development. So like high expression of TEs in early development leads to a high number of de novo insertions in the genome. So there's indeed this, as I just said, so more expression leads to more somatic insertions. And so the final questions we were interested in is, why is there this difference between burst of TE expression between Drosophila miranda and Drosophila pseudoobscura, or why do we think is, am I 2 minutes? Okay, so why is pseudoobscura more toxic? So as I mentioned, in pseudoobscura there's only a burst of expression, but in Miranda there's this sustained increase in expression. So what we did is we looked at heterochromatin profiles very early in development, when there's, in both species we have more expression in males, and later on when in pseudoobscura TEs are sufficiently suppressed in males but not females. What I'm showing you here is a chip, so that on top is Miranda, here's pseudoobscura. The top track just shows you the amount of repeats. So the Y chromosome is highly repetitive, pericentromeres, and for pseudoobscura the Y is highly repetitive in pericentromeres. And in red, I'm showing you heterochromatin for females versus males. And so in both species, you have heterochromatin build up during development at repetitive regions. However, if you look at like this later stage 7, we can see that in Drosophila pseudoobscura, there's very clearly as much heterochromatin on the Y chromosome as there is in the rest of the genome at repetitive regions. But in Drosophila miranda, we actually find that the neo-Y chromosome, even though it's highly repetitive, actually shows very little heterochromatin compared to other repetitive regions. And we can actually like verify that. That's not an artifact by doing like in situ, by really showing the neo-Y chromosome doesn't show a lot of heterochromatin, even though it's highly repetitive. And so what we were thinking, what's going on here is that it is well known that active transcription impedes heterochromatin formation. And as I showed you earlier, the Drosophila miranda neo-Y chromosome is full of actively transcribed genes. So our hypothesis is that this active transcription of genes is causing improper silencing of adjacent transposable elements. And we can test that by looking at heterochromatin enrichment across the neo-Y chromosome and categorizing, like just doing a sliding window approach and looking into windows that either do have actively transcribed genes or not. And indeed, if we have a window that has a zygotically expressed gene, we see less heterochromatin. Conversely, if we look at transposable elements, those that are near zygotic genes, so the blue ones, they show much less heterochromatin than those ones that are far away from zygotic genes. And finally, if we look at transposable elements that have a lot of insertions near expressed near Y genes, they also show much more transcription. So all this data here suggests that there might be an epigenetic conflict between, on one hand, the need to silence transposable elements, but on the other hand, also the necessity to transcribe zygotic genes that are still in the neo-Y chromosome. And so this is like, it brings us up to this idea, and that's actually my last slide about, that there might actually be this, that there might be this intermediate stage of Y chromosome evolutions where Y chromosomes are especially toxic. So early on, Y chromosomes are full of, like beautiful genes that can be transcribed, no repeats. Old Y chromosomes that don't have any useful genes left, they're full of repetitive chunk, but you can actually like easily just epigenetically silence the entire chromosome. But if you have this intermediate stage that Drosophila miranda has, for example, you have a lot of repetitive DNA interspersed with actively transcribed genes, so you can just do this like patchwork of silencing, and you might actually have this very inefficient silence. And then you might actually be much better off to completely epigenetically silence that neo-Y chromosome and dosage compensate the X chromosome in order to avoid this mutational burden that the neo-Y chromosome would be on males, like increased mutation rates and so forth. So as I showed you, Y chromosomes in general can be toxic. We have some evidence that Y chromosomes directly contribute to shorter longevity in males. And we also find that there can be a mutational burden in early development, and especially so if it's a young Y chromosome. And I just wanted to end up thanking the people who did the work, which I already did. Thank you, and we're actually looking for a postdoc in the lab. Thank you very much.